So now we will discuss second part of the protocol of immunohistochemistry, and here we will discuss how tissues and sections they are stained. First of all, there is rehydration of the sections. हम rehydration कैसे करते हैं? उनको हम successively higher concentration of alcohol से lower concentration की तरफ ले जाते हैं. तो हम उनको 100% alcohol, 90%, 80%, 70% इस तरह सक्सेसिवली उनको पास करते हैं और इस तरह से उनकी रिहाइड्रेशन जो होती है. हाइड्रेटेड सेक्शन जो होते हैं, देन दे आर ट्रीटेड विद सम बफर फॉर एंटीजन रिट्रीवल. दिस प्रोसेस इज वेरी इंपॉर्टेंट एज व्हेन tissues are fixed, the antigens or the molecules in the tissues, they may be damaged. They may be modified so that they are not in actually original form. So they are treated with some buffer like sodium buffer so that they are brought in their original form. And this process is antigen retrieval. So rehydration ke baad there is process of antigen retrieval. Then tissues are washed and then blocked with blocking buffer. जाहिर है blocking buffer में हम इसलिए उसको इमर्स करते हैं ताकि जो अनस्पेसिफिक साइट्स होती हैं टिश्यूज़ के अंदर वहाँ हमारी एंटीबॉडीज़ बाइंड ना करें. और जाहिर है इसका पर्पज़ ये होता है कि हम जो हमारे फॉल्स पॉजिटिव रिजल्ट्स होते हैं उनको हम अवॉइड करना चाहते हैं. हमें अदरवाइज फॉल्स पॉजिटिव जो रिजल्ट्स होते हैं वो इंटरफियर कर सकते हैं. After washing, sections are stained with labeled either primary antibody in a buffer or they may be labeled with secondary antibody if the primary antibody is unlabeled. Then samples are washed and the unbound or the antibodies which are free, which are not attached with the respective antigens, with the respective molecules, they are removed. Again, the purpose is to avoid false positive results. Then the samples are mounted in some mounting solution and then finally studied by simple or confocal microscopy. Primary antibodies which are used, they are raised against an antigen of interest present in the sample. So कोई भी, for example, A molecule हम कहते हैं, तो A molecule है तो उसके अगेंस्ट जो एंटीबॉडीज रेज की जाएंगी उनको हम प्राइमरी एंटीबॉडीज कहेंगे. अब इन प्राइमरी एंटीबॉडीज के अगेंस्ट जो एंटीबॉडीज रेज की जाती हैं दे आर नोन एज सेकेंडरी एंटीबॉडीज. सो जाहिर है डायरेक्ट स्टेनिंग में प्राइमरी एंटीबॉडीज लेबल की जाती हैं, इनडायरेक्ट में सेकेंडरी एंटीबॉडीज. सो पर्पज इज that we know what are primary antibodies and what are secondary antibodies. And different fluorescent dyes and substances, they are used to label the antibodies, which may include a fluorophore. It may also include some enzyme like alkaline phosphatase. It may also include colloidal gold. It may also include even radioactive elements. Purpose ये होता है कि different methods से हम अपने जो molecules हैं उनको विजुलाइज कर सकें, उनको हम लोकेट कर सकें विद इन द टिश्यूज एंड सेल्स. दिस स्टेनिंग एज वी हैव डिस्कस्ड मे बी डायरेक्ट और मे बी इनडायरेक्ट एंड दिस वी विल डिस्कस इन द कमिंग स्लाइड्स. सो नाउ डायरेक्ट वर्सेस इनडायरेक्ट स्टेनिंग. डायरेक्ट स्टेनिंग जो है इट इज वन स्टेप स्टेनिंग. आई एम टॉकिंग अबाउट डायरेक्ट स्टेनिंग. इट इज वन स्टेप स्टेनिंग एज वी यूज labeled primary antibody. So labeled primary antibody is used and this labeled antibody is used to detect certain molecules in the cells and in the tissues. Here generally sensitivity is lower. The reason is that there is no signal amplification as only one labeled antibody bind with the respective antigen. Here in the figure it is shown if it is, for example, A molecule in the tissue, anti-A antibody will bind with that particular antigen in the tissue. And this anti-A antibody is labeled with some fluorophore as it can be seen here. So primary antibody which is used here, that is directly labeled, and that makes the antigen which is present in the sample or in the tissues to be visualized. In the indirect staining, we use primary antibody which is unlabeled, and then it is stained with secondary antibody which is labeled one. So primary antibody is unlabeled, and then secondary antibody is used which is labeled one. It is shown in the diagram in the slide. Here again, if there is A antigen present in the cell, this is detected by rabbit anti-A antibody, and then this rabbit anti-A antibody is detected by another antibody which is secondary antibody which is labeled here. This must be raised in some other animal, for example in goat anti-rabbit antibody, and this secondary antibody is we can see here it is labeled one. This is preferred staining as compared with direct staining. The reason is it is more sensitive because there is signal amplification due to binding of many secondary antibodies with one primary antibody. जाहिर है एक प्राइमरी एंटीबॉडी के साथ ज़्यादा सेकेंडरी एंटीबॉडीज बाइंड कर सकती हैं, जिससे सिग्नल एम्पलीफाई हो जाता है, सेंसिटिविटी बढ़ जाती है, और जाहिर है we can see our sample with better sensitivity and with better signal.